thinking about phytochromes, which are funky photoreceptors. So this is literally it changes its shape. It's a shapeshifter. And so it just depends on what quality of light it's going to absorb. And then based on that, it will shift into different forms. And so we're seeing the piece of R form. That means phytochrome R for red light. So that form of phytochrome is designed to absorb red light. It's in the shape that apparently it can absorb red light really well. So that when that happens, it will shift its shape into the piece of FR form. So going back to the piece of R looking like the letter M, when it absorbs that red light, it shifts its form into the Y shape. And then we call it the piece of FR form. Okay, so the phytochrome has shifted its shape.
Is far red light
Just beyond red. Yeah, you got it. Do I have a picture of that? So red light just whenever I talk about this, I always feel, do you all know the YMCA song? Okay, I'm not dating myself on that one. That's what I always think about when I'm te teaching this. But anyways, so once you're in the piece of FR form, now that physical form of the phytochrome is really capable of absorbing far red light. And when it does that, then it toggles back and shifts back into the piece of R form. So it's really quite bizarre. However, it's actually kind of cool because it's just that simple shape shifting that will help the plant figure out if it's in sun versus darkness, how long is it in the sun versus the darkness, and then how it should behave in response to that information. They say that the piece of FR is the active form. What they mean by that is that it is the form of the pigment that tends to go into the nucleus and interact with proteins that either turn on certain genes or turn off certain genes. So it helps to regulate gene expression. Okay. So what the plant is really doing is assessing the ratio of these two forms of phytochrome. How much of piece of R do I have in my body versus how much of the piece FR do I have in my body? And based on that ratio, it will provide important information to the plant about light versus darkness. So the ratio of these two pigments. Okay. Questions on that aspect? Okay, so a couple of other things I added this slide to the slide set that's posted. So you'll see in Educat that it says updated and whatever date I updated it. So this is something I wanted to put in to kind of give some of the nuances of what's happening here. So when you have the sun shining on Earth, during most of the day, the ratio of the red quality of light to far red quality of light is about 1.2 to 1. So what that means is there's a little bit more red light striking the Earth than far red light for most of the day. It kind of shifts a little bit when you get to sunset, then there's a little bit more far red. But anyways, for the plant's perspective, they're getting a little bit more red light than far red light when the light is shining on their body. Okay. Now, generally, when they sense they're getting a lot of red light, they're like, oh, yeah, I'm in sunlight. That's a signal to them that they're in sunlight. So if they are absorbing that red light, if their phytochromes are absorbing that red light, that's actually going to shift their phytochrome to the piece of FR form. All right. So let me go back to, so if they are absorbing red light, if their phytochromes are absorbing that red light, that means that in their tissues, they should have a fair bit of the P-sub-FR form in their body during the day, if they're in full sun, okay? When they are getting more far red light, then that's telling them, oh, I'm being shaded out, or maybe I'm in complete darkness. And if that happens, if they're absorbing more far red light, that means their phytochromes are going to be shifting to the PR form, and they'll have more of the PR form in their body compared to the PFR. So going back, all right, if they are absorbing more far red light, that's going to push the phytochrome into the R form. Make sense? Okay. Now, a couple of other things. If you are a plant, one of the primary things you want to do during the day is photosynthesis. If you're concerned about that, they actually prefer to absorb red light for photosynthesis. Okay, it's more efficient for them. They do absorb some far red light. It's not like they totally ignore far red, but they prefer red light. And that is because the chlorophyll pigment is good at absorbing red light and blue light. All right, we'll save the blue light idea for later when we get into photosynthesis, but chlorophyll is really good at absorbing red and blue light. So chlorophyll prefers red light, if you want to think of it that way, more so than the far red light. Okay, so if you're a plant and your chlorophyll pigments are absorbing that red quality of light, that red wavelength, and using that for photosynthesis, that means that the far red light that they don't absorb mostly passes through the leaves and down maybe onto lower plants or to the forest floor or to the soil in a field. Okay. So this is important to think about as well. So if you have a situation of a forested system that is a complex system where you have different canopy layers, you have an upper canopy of trees, you have a mid canopy with trees that are, lower down, and then of course you have little plants and shrubs on the forest floor area. So different layers, different canopies. So what qualities of light they're getting is going to vary depending on where they are in this forest ecosystem. So if you have the trees in the top canopy, they have full access to the sun hitting their leaves because they're the tallest. And so if you are that plant, if you're a big, beautiful oak tree and you're the tallest in this forest, you're going to want to absorb red light for photosynthesis. So you're going to hog or take a lot of that red light, and you're like, yeah, I don't care so much about far red. I'll let that pass through. And so the far red light starts to go through and strike the leaves lower down. And those plants will absorb some of that far red light, and they're like, oh, okay, I'm getting mostly far red light. And that's telling them I'm in some level of shade if I'm getting more far red light than red. What do you think? Does that make sense? Okay. So depending on where you are in this canopy system of a forest, you will be getting different amounts of red light versus far red light. And that is definitely going to impact not only your photosynthesis, but also your phytochrome ratio. And that can, again, that phytochrome ratio is able to signal to the plant, I'm in full sun, or no, I'm getting partially shaded, or no, I'm getting full shaded. And depending on the plant, they might need to respond to that information and do something different. So for example, what if you are a baby oak seedling and you're like 1 foot tall and you like full sun, but because you're in the lower canopy, you're not getting full on red light because the taller plants are hogging that. They're using it for photosynthesis. So more far red is trickling down to you. And so you're getting that far red, more of that. And so that tells you the oak seedling would be like, huh, I'm getting a lot of far red light, not much red. I'm probably in the shade. Gee, I like full sun. And so they might shift their behavior to put effort into, I need to grow taller, right? I want to get unshaded. In order to get unshaded, I have to grow taller. Or maybe they have to grow and get their branches to the edge of the forest canopy where they can claim some sunlight over that way. So the oak seedling that loves sun is going to shift its gene expression to put effort into grow, grow, grow. We got to get access to sun. But if you are a lower canopy shrub and you're shade tolerant, and that's what you've adapted to over all these millions of years of evolution, you might get the same signal that the oak did right next to you, and you're like, well, I don't care that I'm getting shaded out. I'm fine with that. I've adapted to that. I can grow just fine. Right? So it's not like if you're shaded out, you must grow. I'm not trying to say that. It's just if you're a sun-loving plant, yes, you would shift your behavior. I must grow. But if you're a shade tolerant plant and you get the signal, I'm getting shaded, it's like, yeah, okay, fine. Right? Does that make sense? Okay. So the ratio of phytochromes really can provide a lot of information and when you think about more complex ecosystems, there's a lot of dynamic going on there and a lot of decisions, so to speak, different plants will be making based on whether or not they have more red light or far red light striking their body, and that will shift their phytochromes ratios as well. So here's a thought experiment, and you can actually do this in the lab, and it works. They've done it many times, and I think the scientists have fun with these types of experiments. So here is an example of a plant that they call a long day plant. So what that means is that it has to go through a light period that's longer than some critical length of time. All right. So that's how you interpret this drawing. That dashed line is your critical length of time that the sun has to be up longer than that in order for the plant to go ahead and flower. All right, so it's a long day, or you could think of it as a short night plant, either or. So you can see that the criteria has been met. This plant was in light beyond that critical period. And so that gave the information to the plant to go ahead and flower because it's a long day plant. Then if you have the same plant and you create a situation where it didn't meet that critical period, it's just shy of that, right? It went into the dark just shy of that. So it didn't go past that critical length. So it's not going to flower. And you can experimentally do this and it works every time. Okay, so let's go back. So if that plant was exposed to lots of day length, lots of sunlight through that critical period, and we know sun has a little bit more red light. So what that's saying is that that plant's phytochromes actually were there's more of the piece of FR form in its body because it was getting plenty of red light over that critical period. Okay. And that told the plant, go ahead and flower. All right. When it didn't meet that critical period, then what happened was you started to see a buildup of PSABR in its tissues. And that was actually telling the plant, I'm not in the sun long enough. I'm in more darkness than I should be to flower, so I'm not going to flower. Okay. Now they've done these types of experiments where they repeat the middle experiment where they don't put the plant in the sun or in the light for a long enough period. So you would say, well, it shouldn't flower then because it's a long day plant. But then they came in the middle of the night, probably paid a grad student to do this, and turned on some red light onto the plant. So that's being shown there. A little bit of red light, a flash of red light. And when they've done this experimentally, like just a few seconds, 10 seconds or less is enough to cause this reaction. So you see that when they came in the dead of the night, flashed that plant with red light, it flowered. And so the way to interpret this is that the plant is really behaving only to the last quality of light it was exposed to. Right? So it's almost as if the plant thinks it was exposed to light this whole time. Because remember, light has a lot of red quality to it. You know, there's more red light than far red light. So you can either flash pure red light on it or just a bright, normal light, and that has more red than far red. And it tricked the plant into thinking it's time to flower. Kind of weird. So what happened to the phytochrome pigments? So let's think about that. When it got hit with red light like that, what was the ratio of the phytochrome pigments in that plant? So if it's absorbing quite a bit of red light for that 10-second burst, which phytochrome form will be more abundant in the plant?
P sub FR.
Yeah, you got it. P sub FR. So if it's absorbing that red light, it's going to go and shift the phytochrome to the P sub FR. And that's the active form. It's like, oh yeah, go ahead, flower. So you can do these experiments over and over, and you can keep flashing light at these poor plants, red versus far red, and you can trick the plant into go ahead, flower, or nope, don't flower, just by hitting it with these different qualities of light, which is really impacting their phytochrome ratios. Okay. So in this next example here, someone came in, hit it with red light for 10 seconds, waited maybe a half hour, and then came back and hit it with far red light. And so if they hit it with far red light, then that means they're going to have more P sub R in their body compared to the PFR. And that's telling them, I'm in the dark, or I'm getting really shaded. It's not time for me to flower. I didn't hit my long day, so I'm not going to flower. Does that make sense? Okay. So whatever quality it saw last is going to shift the phytochrome ratio and will tell the plant either go ahead and flower or don't flower, depending on what the last quality of light.
If you're looking at the one on the very left, and then at night you hit it with a whole bunch of the far red light, would it still doing? Because it's getting like enough daylight, or
So if you take this one here
yeah,
and you come in and hit it with far red,
but it's already getting like more light than it needs
yeah.
to hit.
And that could mess with it. I think that would mess with it. I think it could abort, like stop the process of flowering. I don't know that they've done those experiments. Somebody must have done that experiment because I would think that would, if they did a comparable experiment with a control, I would guess there'd be less flowers. I think some flowers might So developmentally, once they kick into that, it's kind of like go, go, go. And so they might have some flowers, but then if some of the flowers that are still kind of young in their growth might abort, stop.
Mhm.
I think that would, but that's a really good question. Because most of the studies I've read, they don't try it. So I would think somebody would have tried it.
Mhm.
If I can find any information on that, I'll let you know. Okay. So really this red, far red, playing around with that, what their scientists were really doing was messing with the phytochrome ratio. And that phytochrome ratio was telling the plant how long they were in the sun versus how long they were in shade or darkness. And then they had to decide what to do with that information. And in this particular case, they're looking at should I flower or not? So, so basically, when you look at especially plants in natural settings, not in an experimental lab, but when they're out living their life in the real world, they really are paying attention to this ratio of phytochrome in their tissues. And through that information, that allows them to tell how long they've been in the dark versus the night, or the sun versus the darkness. And it also lets them know if they're getting shaded out by other trees nearby, for example, and what they should do about that. Oh, I already had that in this slide. All right, so I put that in the previous slide too, but I thought that was important to note that sunlight has a little bit more red light. So when you think about this ecosystem, where there's multiple layers, it's not just phytochromes you have to think about because, of course, one of the huge things that plants are doing during the day is photosynthesis. So you also have to appreciate that they really want the red light. They prefer the red light versus far red for the photosynthesis, and that can impact plants further down below those higher up plants like we were talking about. That oak seedling is going to hog most of the red light and that allows the far red light to just kind of pass through, and the lower plants have to make do with the more far red light. So the tallest trees really, they're the ones that are adapted to full sunlight. They can hog as much red light as they need, and they'll let the far red light go through. And so when you think about more complex ecosystems, all the plants, so they might be in the same relative area, they're getting very different signals because of their phytochromes, what that ratio is. So next time you're out hiking in the woods, hopefully you can appreciate that that's going on. Although, well, evergreens, even though it's cold out on sunny days, and if it's warm enough and it's sunny out, or even partially cloudy, evergreens will, under the right conditions, go through photosynthesis and do some growth. The deciduous trees, they're all sleeping right now, but okay. Any questions lingering about phytochromes? It's a weird concept. I remember the first time I learned it. It's just weird. So spend some time studying it, please, and thinking about these two forms and the qualities of light. Which reminds me, so today's Friday, right? So a week from today will be the first lecture exam. And phytochrome will be on it, of course. So make sure that you're studying a little bit every day. And then at the end of the week, you should be reviewing your week's notes. How do you all like the library, by the way, for studying? Eh?
Very bland in there.
I've been hearing that from so many students.
You're walking.
Yes,
Yeah.
someone used the word sterile. It's so sterile in there. I agree. Oh, although I actually haven't gone up. There's a fireplace? somewhere?
Yeah, it's nice. It's upstairs.
Is it like a fancy, like,
It's like a dual-sided
oh,
fireplace.
yeah, it's like you see in a hotel, the
Yeah.
lobby or something, yeah. Not like a real wood, of course, they wouldn't let us do a wood stove, but
They made the tables 6 inches off the ground.
again,
Yeah.
they did that in our atrium in Weston Hall. It's like people don't study hunched over, you know? Okay, that's really weird that they didn't learn from that experience. I know they're going to make it less sterile, um, but I guess they can't touch anything they can't put anything on the walls until the final inspection, and which hasn't happened yet, I guess. And so I know they plan to get more plants in there. Yay. So that's good. And then I know they're going to be doing more stuff with the walls. But one of the things I noticed when you walk through the main lobby area, those study rooms right there, big glass, looking out to, I mean, how can you study with people traipsing by you? Who thought of that one? There's so many rooms like that where there's these big windows leading out to people. I was like, how can you study when you have all that going on? And then people are going to look in there. So you're getting looked at,
Yeah.
It's like, I would feel like a fish in a fishbowl. I would not like that myself. I'm hoping they're going to put curtains or some kind of, I don't know.
It's too.
Yes.
The crossing in
It,
between.
it gets so
It's
messy.
bad.
It's bad. I know. I feel sorry for the maintenance people. So, yeah, I'm hoping it will improve, but I've heard almost everyone I've talked to says that it looks just so sterile. So, hopefully they'll make it better. I just can't. I just shake my head with all those windows though. It's like I don't get it. I don't get it.
Are they moving the plants from the bio building to?
Yes, they already have, I think. Are there plants in there currently? I haven't really. But they plan to get more. So hopefully they have a budget for that. They can buy more. I'm hoping they're not relying on us to provide it because we have to fill up our atrium again. We've been given orders by Dr. Leonard, now that the library plants are out of the atrium, we need to fill it back up. So we're going to try to do that. Okay. I did want to spend some time getting into the next slide set, which is getting into some um, photosynthesis ideas. So I know some of you have had Plant Fizz, and this is going to be a little different focus. So in an intro botany course, what we tend to focus on is a little bit of the light reactions, get you just a little bit of that, but we focus a little bit more on what I call part two of photosynthesis, which is how do they make their sugars? Because if you can understand that, if this is the only plant class you'll ever take, and if you can understand part two of photosynthesis, you'll have a pretty good idea of when you're out in different habitats, and hopefully if you get a chance to travel to different habitats, you'll understand why certain plants grow in certain areas of the world and not others. Because how they do part two of photosynthesis really does drive how well they can adapt to different habitats or not. And of course, for crop plants, that's important too, because with global climate, well, it's important for any plant with climate change going on, that's going to be shifting, the habitat of plants, and that could profoundly impact how well they do photosynthesis, especially part two of photosynthesis, with the climate change impacts that will be felt by these plants over time. So that's why we tend to focus more on part two. When you think about photosynthesis in the big picture, it's basically their way of getting a lot of ATP through the light reactions. And then they're going to use a lot of that ATP to make their sugars. Okay. But first they have to get that ATP made. So part one is this idea of they need to use sunlight energy in order to get their ATP. And so we're going to zoom into the chloroplasts for that. And in particular, the chlorophyll pigment is the workhorse. It's the chlorophyll pigment that will be absorbing light energy, preferentially red and blue. It prefers that pigment, really prefers, it really is adapted well. When I say prefer, what I mean is it's adapted to maximally absorb red and blue light. Doesn't care so much about the other qualities of light. So when you are learning about photosynthesis, you often get this equation put out to you where the plant will be taking in CO2 and then in the presence of water and in the presence of light and with the chlorophyll's pigment help, you're going to end up with C6H12O6. The plant also makes oxygen and water. So this is what I call the big picture idea. This encases both parts of photosynthesis, the light reaction, and part two is, so what I call the light reaction, that's part one of photosynthesis. Part two is making sugars. We often call that the biochemical reactions. And so this encompasses both of that. There's a lot going on in order for this to happen. So we're going to get into some of the details of this idea. So another thing I want to point out is when I say to you C6H12O6, what sugar do you typically think about?
Glucose.
Glucose, which is fair. That's what I think about when I hear C6H12O6. If you've taken chemistry classes, we usually think about glucose for that. However, I want to point out very emphatically, plants do not make glucose as their sugar. Okay, that's not what they're going for. Okay, so this is a little bit, I understand why they write it out this way, but in Plant Phys, if you've taken that class with me, you know that I don't really like this equation so much because it makes you think about glucose, and glucose really isn't the sugar that the plant is going for. The plant is actually going for sucrose. Okay, that's the one type of sugar. Now, sucrose is made up of glucose, but it's attached to a fructose. So when you look at a sucrose molecule, it's a disaccharide, which means it has two parts to it, glucose and fructose bonded together. So there is glucose in there, but the plant really wants to make sucrose. That's its goal, not just glucose. It's going for sucrose. So please try to keep that in mind. So here's a chloroplast, and they're showing both parts of photosynthesis. So the photochemical reactions or the light reactions, I just say light reactions, but photochemical is accurate. So that's where the chlorophyll embedded inside the thylakoid membranes inside the chloroplast. They're absorbing that light energy. And as they do that, you get some ATP being made, you get some NADPH being made. Both of those are very important for part two, which we call the biochemical reactions. Okay, part two, which is to make sugars. And really, they're going for sucrose. So part two is this idea of taking in the CO2. So how do you think the plant brings in CO2? What structure are they using for that? Yep, stomata. Okay, so they have to have their stomata open for this to get the CO2 brought in. And then you see the CO2 goes into this Calvin cycle. And through that process, you do get some sugars made. Okay, so this is putting it all together, part one and part two. Here's another way to look at it. And here I've written the chemical equation that I use in Plant Phys, which is a little bit less confusing and what I think is more appropriate, where instead of saying C6H12O6, you simplify the equation down. You get rid of all the numbers essentially. And when you do that, you're left with CH2O, which is the basic building block of a carbohydrate or sugar. Okay, it's just a basic building block. And so that way you get away from thinking about glucose. Okay. So this is what I show in Plant Phys. It's just another image showing how these two parts of photosynthesis are linked in the sense that the products of the light reaction, ATP and NADPH, those are the products of the light reaction, feed right in to the biochemical reactions or the carbon reactions, kind of like that, better, which is the Calvin cycle. Okay. And once the Calvin cycle runs, you do end up with some sugars. And it's actually sucrose that the plant will build. That's the one it cares about. So now we're going back to the idea of the chloroplast organelle. So remember, it's got two membranes. It's got that outer membrane and inner membrane that surround it. And then there's that third membrane inside. So there's an internal membrane. The whole thing is called the thylakoid. Okay, the whole internal membrane is called the thylakoid. And then the watery portion that the thylakoid is floating around in, we call that the stroma. Okay, so you've had all this. It's just been a week or two. So it's that thylakoid where everything's happening for the light reactions. Okay, so that's why it's all green, the thylakoid, because all the thylakoid membrane has a bunch of chlorophyll pigments embedded in it. And if you were to cut open some of the thylakoid, that's what they're showing in this right image. If you were to cut open that membrane and look inside, we call that region inside the thylakoid the lumen, right? The lumen. You have lumen when you're in your gastrointestinal tract, all the inside of your intestine. We call that the lumen. So it's not anything unique to the chloroplast. It's just denoting the internal space here of a membrane enclosed area. Okay, so all this is happening inside the chloroplast in that thylakoid membrane. So with the light reactions, the plant has this amazing ability to take water and strip electrons off of it. That is incredibly difficult to do. And that's why they need light energy. Part of the light energy they absorb allows for the plant to hit water with enough energy to strip electrons off of it. That's a phenomenal process. And water loves to hang on to its electrons. It does not want to give those up. And so that's why you have to absorb that light energy. So through that action of absorbing light energy, electrons are actually stripped off of water. And the other thing that happens, once you start stripping electrons off of H2O, you get left with, of course, electrons are getting stripped off, but you also get left with H plus or protons. And of course, oxygen will get released too. So I'm sure if you had the intro to photosynthesis at some point, either in BI 111 or maybe in high school science, they talk about oxygen is one of the products of photosynthesis, which is true. And we sure care about it because that allowed us to evolve here on Earth. However, that's not really what we focus on when we teach photosynthesis because it's not really important for what we think about when we think about what is the goal of photosynthesis. Certainly, the plant wants those electrons, and I'll explain a little bit more why. And it definitely wants those protons. That's very important to the plant. But the oxygen it just releases out of its stomata and like, don't really care too much about that. It's just we care a lot about it, so we often mention it. But we're not going to focus really on the oxygen. We're going to think a lot more about the electrons and the protons. So when we see that water is getting split, it's actually in that lumen portion inside the thylakoid membrane, in the hollow portion inside. Water's actually in there getting electrons stripped off of it. All right. And then those electrons are going to pass along an electron transport chain or system. As electrons are passing down this electron transport chain, it creates a situation that allows for ATP to get produced. And I'll show you what I mean by that. we actually see ATP being produced not in the lumen, but in the stroma portion. So that's a good thing because if ATP is being produced in the stroma, it's set up and needed for the biochemical reactions, for the Calvin cycle, so that the plant can make its sugars. It needs that ATP for that. The other thing that's happening as those electrons are getting passed down that chain is that there will be like an ultimate electron acceptor where the electrons will go and form a chemical, and that is NADP plus is the final electron acceptor. It's sitting at the end of the chain. Once it gets those electrons, it will get, we say it's reduced. So when a molecule accepts electrons, then we say it's becoming reduced. And when it does accept those electrons, it also grabs a proton and forms the NADPH molecule. And that's also happening in the stroma. So both ATP and NADPH are being produced in the stroma through the light reaction process. And that NADPH is really important. You know, we already said ATP is really important, but so is the NADPH because that's also needed to make sugars. So it's also needed for part 2. Okay. So what we just said in words is being shown here visually. I'm not going into huge detail on this like I do with Plant Phys. Plant Phys, we do dissect this and go through it very carefully. However, there are some major steps that I want you to be aware of. So you see here that there are, this is the thylakoid membrane that they're showing. This is the thylakoid membrane. This is the inner portion of the membranes. This is the lumen portion. And then the watery portion of the chloroplast is called the stroma. And you can see in that thylakoid membrane that there are things labeled PS2 and PS1, and they're colored green. Okay, so this is where the chlorophyll is. Chlorophyll's in PS stands for photosystem. Okay, so photosystem 2, photosystem 1, lots of chlorophyll is present in those two photosystems. Hang on just a second. So PS stands for photosystem. I don't know if I can. That's what the abbreviation is. They named it 1 and 2 because just in order of discovery. So they discovered photosystem 1 first, and then they, oh yeah, there's another one. So they called it photosystem 2. All right. And what they're showing here is this idea of electron flow by the black arrows. And they're showing that it starts with water over here. So what's happening is when photosystem 2, which has a lot of chlorophyll in it, absorbs light energy, it allows for electrons to be stripped off of water. And then they're showing the electron here in the blue. So what they're trying to show is, yep, you got electrons being stripped off of water, and then it's going to be sent through this electron transport chain process all the way to NADP plus. That's the final electron acceptor. Okay. And so you can see there's several players in the game here. You have this PQ molecule that's going to get the electrons. It's going to get then shared with the cytochrome B6F and then to the plastocyanin. You don't need to know all the words. And then to photosystem 1. You need to know there's multiple players in addition to photosystem 2 and photosystem 1. You see that both photosystems are getting hit with light energy. There's chlorophyll in both of those. And both of them do need to get hit with light energy at pretty much the same amount in order for this to keep going nicely and not kind of go into chaos. All right. So you have this electron flow from water. The electrons will get passed on to the electron transport chain all the way to NADP plus. And then when NADP plus gets that electron, it gets converted to NADPH. Have any of you, let's see, in BI 111 or maybe another phys class, do they talk about respiration, like what's going on in our mitochondria with the electron transport chain? Okay, so if you've had that information, this should sound vaguely familiar. It's just that it's the chloroplast, not the mitochondria. But it's the same basic idea that, an electron transport chain process is not unique to chloroplasts. Our mitochondria are doing it right now. Microbes do this in their cell membrane, well, some microbes do photosynthesis, but the electron transport chain idea, it's a mechanism that has evolved over time and has been put to different uses, so to speak, in the sense of this one's being used for in the photosynthetic arena, whereas our mitochondria, it is where we have our oxygen sitting as the final electron acceptor instead of NADP plus. But the end goal is wonderful because you get that ATP, whether it's in your mitochondria or here in the chloroplast. You generate a lot of ATP, which the plant needs, just as we do, except they're using a lot of their ATP to make sugars. Wouldn't it be awesome if we could just make our own food? Just go out in the sun for a couple of times each day and we're set. Think how much time we'd save not having to cook dinner. I'm getting tired of cooking dinner during the semesters. Uh, so Okay, so we have NADPH and ATP by the end. So we have to think a little bit about how that ATP is getting made. I think I was able to show you how the electron flow finally ends up at NADP plus, so you do get that NADPH. But we have to think a little bit about, okay, they're also showing ATP getting made, and we have to think about that. But we ran out of time. So I'll leave you hanging on that. We'll get into that on Monday. Reminder, the lab quiz homework is opening at 1 today. Okay, 1 today, and you have until Wednesday at 11 to do it. Okay.